Yes, so the next speaker is my dear colleague Dr. Jung from Korea. Yes, he's a known researcher. We all know him now very well, and he's very motivated and an interesting person. He will talk about an identification of potential therapeutics for vitiligo by mass screening for medicines in a Korean database. So please go ahead.
It's okay?
Yeah, looks good.
Yeah, thank you. Yeah, thank you for introducing me. Hello, everyone. My name is Jung Bae from the Catholic University of Korea. It is a great honor to be here today and share my recent work on big data. The development of new drugs is an extremely lengthy and costly process. Drug repositioning is an approach to identify new use for approved drugs outside of their original medical indications. It could be used as an alternative strategy for identifying new indications of in-market drugs in areas with little industry interest, such as vitiligo. A recent review on drug repositioning outlines 8 possible strategies. Among them, retrospective clinical analysis using electronic health records would be a viable option in the field of vitiligo. Each patient visit is stored in a collective database that then makes this information available to explore the behavior of patients. The electronic health record includes assigned diagnosis, demographics, treatments, and medications. Using healthcare big data, we can do a variety of researches, such as comorbidity studies, patient clustering, machine learning, and various core studies. At this time, I would like to present my recent work to attempt drug repositioning for patients with vitiligo with the database. Korean National Health Insurance covers up to 98% of the 50 million people who live in Korea. It is one of the largest NHI systems in the world. It provides all insurance claims information in Korea for over 10 years. This is a pilot study using the database. My question was, are there any drugs that could prevent the development of vitiligo? In the literature, I found some case reports on the darkening of white hair following levodopa therapy in patients with Parkinson's disease. The authors described that levodopa could induce melanocyte regeneration in some patients. More surprisingly, long ago in the Lancet, 1973, some authors tried the use of levodopa in 7 patients with vitiligo and found some degree of repigmentation in 4 patients, and 2 showed almost complete repigmentation. We hypothesized that levodopa could be beneficial for vitiligo patients. We designed a case control study to explore the association of levodopa use and development of vitiligo. We identified all vitiligo patients over 40 years old in Korea and set up a propensity score matched controls. We searched the past history of levodopa use before the index day. We found that the risk of vitiligo was negatively associated with the use of levodopa, odds ratio of 0.801, and the association was more remarkable with the use of levodopa for more than 6 months with an odds ratio of 0.698. This study would be an example. The next step was try a more systematic approach using an automated method. The first step was to list all systematic drugs in the NHI database. That means all drugs prescribed in Korea. Second, we conducted sequential case control studies to find drugs that had a negative correlation with vitiligo development. The next was to verify the effectiveness of each drug through sequential code study of each drug, user code and non-user code for the drugs selected in the previous step. I'll explain the final step later. A total of 43,553 main ingredients were registered in the Korean NHI system. Among them, we finally selected 1,732 main ingredients used as systemic drugs as candidate. Next, the mass screening step. These are demographics. Both groups are well matched to age, sex, and insurance type. A total of 1,732 case control studies were conducted on these 1,732 principal ingredients. We examined the history of each drug used in both groups to find drugs that negatively correlate with the development of vitiligo. In total, 21 out of 1,732 drugs were identified as less prescribed in patients with vitiligo compared to the controls. Here is a list of 21 drugs, blah, blah, blah. Then, using a sample cohort of 1 million people, we created a drug user cohort and a propensity score matched non-user cohort for each drug to investigate the occurrence of vitiligo in the both groups. A total of 21 cohort pairs were created for 21 drugs, and 21 cohort studies were conducted. For each drug, a drug user and non-user cohort were created, and we compared the incidence of vitiligo between the two groups. We repeated this type of cohort study for each of the 21 drugs. Through the 21 cohort studies, we found two drug user groups that have significantly reduced incidence of vitiligo. That was glimepiride and metformin. The hazard ratio was 0.807 and 0.799 respectively. Our study has some limitations. Diabetic patients who take both drugs at the same time, protective effect of diabetes itself, decreased medical availability in severely ill patients. It could have a selection bias. To solve this problem, we performed post hoc analysis using the diabetes cohort of 92,496 patients with diabetes. The first analysis was to assess the effect of glimepiride use. In univariable analysis, the hazard ratio was 0.514. However, after adjusting for some variables, it lost the significance. Next is the effect of metformin use. In univariable analysis, the hazard ratio was 0.299. After full adjusting other variables, the use of metformin was still significant. The hazard ratio was 0.492. In total, 21 out of 1,732 drugs screened were identified less prescribed in patients with vitiligo compared to the controls before the diagnosis of vitiligo. Among them, the incidence of vitiligo was significantly lower in individuals with exposure to metformin and glimepiride. In the diabetic cohort, exposure to metformin was associated with lower risk of instant vitiligo, hazard ratio 0.492. In conclusion, a potential protective effect of metformin for vitiligo was suggested from our mass screening approach. Metformin could be attempted for its potential application as a therapeutic or preventive agent for vitiligo. Our approach could be an alternative strategy for identifying new indications of in-market drugs for rare disease. Thank you for your attention.
Okay, great. Thank you for this presentation. It's a great illustration of the power of linked databases. I had a question for you in terms of trying to interpret the hazard ratio. Let's say, what would be the hazard ratio for a drug that we actually know does work? So do you have any, because we're trying to figure out how to interpret, it's either significant or not significant, but also the magnitude of that hazard ratio. Is there any way that you can estimate how that hazard ratio would be for tacrolimus, for example?
Actually, in the cohort study, we set up the drug user cohort and non-user cohort who doesn't have any history of vitiligo before the drug taking, and then we evaluate the instance of new vitiligo over the two cohort, and we compare the instance of vitiligo using the hazard ratio.
Okay. So a couple of questions that come up from the chat. Did you with levodopa, was there any signal in the, your automated search when you looked at the levodopa?
Yeah, actually, at the screening phase, I found levodopa is lower as a negative correlation with vitiligo development, and then the next in the validation phase, I think my 1 million population cohort is a little bit too small to find the levodopa user. So I think if I have more big size of a population, and then I can do more research on the levodopa.
Okay, and just one additional comment or question from Professor Ezzedine. This is a brilliant study. I only have concern about performing the propensity score and the choice of variables for this.
Actually, it's very difficult to adjust all kind of variables. So we use age and sex and Charlson comorbidity index to set up the propensity score matching.
Okay, thank you very much.